staining in histology usually comes from a combination of various histological dyes. The most common combination being the hematoxylin-eosin, where the hematoxylin is the basic component responsible for dark purple to dark blue, dark violet stain, and the eosin, being the acidic component, is responsible for the reddish to pink tones, and the combination of both gives the whole palette of histological stains. Let's just provide some examples. I will start with the strongly eosinophilic structures, such as the contractile filaments in cardiac muscle cells. So the contractile myofilaments that are responsible for the cross striation are eosinophilic with quite high intensity. Okay, this is eosinophilic. With high intensity, and this would be a cardiac myocyte. Medium intensity could be found, for example, in enterocytes. That's the epithelium lining the intestine. Or cells with numerous mitochondria, such as the epithelium of the liver, so-called hepatocytes. We can't see individual mitochondria in the routine microscopes, but we can see the eosinophilic staining that is due to the presence of numerous mitochondria. So that would be enterocyte, epithelium of the small or large intestine. That would be a hepatocyte, with many mitochondria. Let's proceed to highly basophilic structures. A good example would be plasma cell, the one with the nucleus that has a typical pattern of chromatin, cartwheel or clock face chromatin. But in this point we focus on the rough endoplasmic reticulum. That's the granular endoplasmic reticulum with many ribosomes. So except for this region close to the nucleus, you can see the granular endoplasmic reticulum. This is strongly basophilic staining, so I would call it basophilic with high intensity. So it's a plasma cell. This is a cell that produces immunoglobulins. Another example of basophilic cell, but this time with medium intensity, could be, for example, the cell of membrana granulosa, which is one of the layers of the growing ovarian follicle. Right? medium intensity, and that would be membrana granulosa cell of the ovarian follicle. In reality, there is a whole palette of various types of stain. These were some examples. Let's consider also cell and cytoplasmic inclusions. They might also contribute to the staining of cells. I will start with lipid droplets. And a good example for this would be fat cells. This is a young fat cell where the lipid droplet, there are multiple lipid droplets distributed in the cytoplasm, and the fully differentiated mature fat cell where the lipid droplets have fused into a single large lipid droplet. Another example could be some cells producing steroid hormones. They also have a tendency to accumulate lipid droplets with cholesterol, because cholesterol is used for synthesis of steroid hormones. So that would be a young fat cell called adipoblast. That will be an adult adipocyte. And this would be a steroid producing cell. Such as the epithelium of the zona fasciculata, that's one of the layers of adrenal cortex. And what these cells have in common, they have lipid droplets. Here it's mainly cholesterol. Here there could be various triglycerides. These lipid droplets appear as optically empty white regions in the routine sections because the lipids have been dissolved during histological processing. Another example of inclusions could be glycogen granules, such as occur in the vaginal epithelium. So if this would be one of the epithelial cells lining the mucosa of vagina, the cytoplasm looks nearly whitish. Because the glycogen does not stain either with hematoxylin or with eosin. So it's light cytoplasm due to that, as a consequence. So this is vaginal epithelium. During the reproduction period, where the cells are sensitive to estrogens, resulting in accumulation of glycogen. There could be hepatocytes, these are other cells that accumulate glycogen. Skeletal and cardiac muscle tissue also accumulates glycogen. And the endometrium, that's the lining of the uterus during the pregnancy, is called decidua. it also accumulates glycogen as a source of energy after the implantation of the embryo. Let's go to other inclusions, pigments. You can classify pigments as endogenous, that are products of the metabolism, or a separate group would be hematogenic pigments that are the hemoglobin breakdown products, and there could be more pigments like exogenous pigments. The common endogenous pigment would be melanin. It occurs in forms of melanosomes, which are pigment bodies produced in cells called melanocytes, are released to extracellular space and accumulated in adjacent cells, such as cells of the epidermis. So if this will be a pigment cell producing the melanosomes, these pigment bodies, and they will be accumulated by adjacent cells called keratinocytes of the epidermis of human skin. Melanin could occur also in the form of so-called neuromelanin in nervous tissue. So if I have an example of a neuron, this will be a cell body and the axon of some neuron. Then some neurons in the human brain have a tendency to accumulate melanin granules, and this time we call it neuromelanin. And this is a neuron. Now, which neurons are doing that? For example, these are the neurons of the substantia nigra in the mesencephalon, or the neurons of a nucleus called locus cerebrus. Also, retina has pigment epithelium. It is a shape of one layer of cuboidal cells and the cells are accumulating melanin as well, so that the pigment epithelium of retina, the outer layer to which the photoreceptors are diverted. So that would be pigment layer of retina. So that's the melanin pigment. Another pigment that occurs in the human body is called lipofuscin. It's a residual pigment that is accumulated in cells of elderly person. For example, in cardiac myocytes that are long-living cells, you can find on the poles of the cell nucleus, you can find such yellowish, yellow to brown granules. That's lipofuscin. So this will be the cross striation of the cardiac myocyte. And as there is a nickname for lipofuscin, it's called wear and tear pigment. And it represents residues after incomplete lysis and digestions of various lipids and lipoproteins, and it's accumulated in elderly cells. That's why wear and tear pigment. An example of hematogenic pigments that come from the breakdown of hemoglobin is hemosiderin. And the hemosiderin could be found, for example, in macrophages that are dealing with dead red blood cells. So these are macrophages, and they have these hemosiderin particles. These macrophages are even called siderophages, which is a macrophage with hemosiderin that occurs in, for example, in the red pulp of spleen. Of course, in the red splenic pulp, that's where most of the red blood cells are dying, and their hemoglobin is decomposed into many molecules such as hemosiderin, or in bone marrow macrophages.